Hello, this is Amy Perrin, and I have been studying the formation of topographic maps in the brain. This particular paper is by Wang et al. From 1998 and is about the formation of topographic map of olfactory sensory information in the olfactory bulb. Before I begin, I would like to give a brief overview of olfaction. In the roof of each nostril is a region called the nasal mucosa. This region contains the olfactory epithelium. The area of this olfactory region is about the size of a postage stamp in humans. The epithelium contains sensory cells as well as Bowman's glands that secrete the mucus that bathes the surface of the receptors. This is an aqueous secretion containing mucopolysaccharides, immunoglobulins, proteins, and various enzymes. Finally, the nasal epithelium contains the receptor cells, numbering to about 10 million in humans. They possess a terminal enlargement that projects above the epithelial surface, from which extend 8 to 20 olfactory cilia. These cilia are what contain the smell receptors. Now, let's move on to the paper. Most sensory systems spatially segregate afferent input from primary sensory neurons to construct a topographic map that defines the location of a sensory stimulus within the environment, as well as the quality of the stimulus itself. Olfactory sensory processing does not extract spatial features of the odorant stimulus. Relieved of the requirement to map the position of an olfactory stimulus in space, the olfactory system employs spatial segregation of sensory input to encode the quality of an odorant. The topographic map revealed in the olfactory system differs in quality from the orderly representation inherent in the retinotopic, tonotopic, or somatotopic sensory maps. The olfactory system does not maintain an orderly representation of the receptor cells in the periphery. Olfactory neurons differ from one another by nature of the receptor that they possess, not by their location on a receptor sheet. Individual olfactory sensory neurons express only one of the 1,000 receptor genes, such that the neurons are functionally distinct. Cells expressing a given receptor in the olfactory epithelium are randomly dispersed within one of 4 broad zones. Sensory neurons extend a single unbranched axon to the olfactory bulb, such that the projections from neurons expressing a specific receptor converge on 2 out of the 1,800 glomeruli. The pattern of convergence is absolute and invariant in all individuals in a species. The bulb therefore provides a spatial map that identifies which of the numerous receptors have been activated within the sensory epithelium, such that the quality of an olfactory stimulus would be encoded by specific combinations of glomeruli activated by a given odorant. So, what causes this convergence? The researchers found that deletions or nonsense mutations in the P2 odorant receptor gene cause the axons of the cells to wander rather than converge on a specific glomerulus. Here is a schematic representation of the gene targeting strategy used to delete the P2 receptor coding sequence. Homologous recombination between the targeting vector and the wild-type P2 gene results in the replacement of the P2 coding sequence. Transient transfection with the plasmid expressing the Cre recombinase results in the excision of the TK-neo cassette. Now, these are the results of the P2 deletion experiment. Pictures are a whole mount view of the nasal cavity and medial aspect of the olfactory bulb. The top row of images are time-lapsed over a series of weeks from the P2 mutant mice. As you see, the axons project diffusely into the olfactory bulb and do not converge on a glomerulus. On the bottom row, in contrast, is a P2 wild type. Here, convergence at a single glomerulus is apparent. The researchers, in addition to this deletion experiment, performed a series of receptor substitution experiments that, for the sake of time, I will not discuss. After considering all the research, the data strongly suggest that the odorant receptor plays an instructive role in the guidance process of the axons from the sensory cells to the specific glomerulus. So to recap, in the olfactory system, neurons expressing a given odorant receptor project with precision to 2 out of the 1,800 glomeruli within the olfactory bulb. Since the positions of individual glomeruli are topographically defined, the bulb provides a spatial map that identifies which of the receptors have been activated in the sensory epithelium. Therefore, the quality of the stimulus would be encoded by the specific combination of glomeruli activated by a given odorant. This topographic map encoding odor quality in the bulb has been shown to develop without apparent contribution from activity-dependent processes. Mice lacking the olfactory cyclic nucleotide-gated ion channel fail to exhibit odor-evoked electrophysiological responses in the sensory epithelium, but the pattern of convergence of like axons in the bulb is unaltered in these mutant mice, arguing strongly that olfactory experience is not necessary for the establishment or refinement of the topographic map. These findings, however, do not exclude a role for activity-dependent processes in the maintenance or potential plasticity of the map after it is established. For example, neurons that do not express functional odorant receptors have been observed to have a significantly shorter half-life. Activity-dependent processes may therefore be essential for the survival of the olfactory neurons postnatally and, in that matter, may alter the input to individual glomeruli, altering the sensitivity to individual odors. To summarize, two main takeaway points from this article are: an olfactory neuron is given a distinct identity by virtue of the receptor that it expresses, and this receptor dictates the pattern of the projections in the olfactory bulb. I found this article very interesting and helpful when it came to my research in sensory topographic maps in the brain. Of all the sensory maps, the olfactory map is one of the least studied so far, but more research is being done. One of the main questions I would like to have answered in the field is, how does an olfactory sensory neuron choose to express 1 out of 1,000 possible odorant receptors? I think it's fascinating that a cell can choose 1 out of the 1,000 and hope that this question is soon answered.